So for over 2 decades now, billions of dollars have basically been poured into treating Alzheimer's disease as a purely biological problem.
Right, yeah. It's always biology first.
Exactly. I mean, we've all seen the headlines, right? Rogue proteins, toxic plaques, neurofibrillary tangles.
Oh, absolutely. The narrative is everywhere.
Yeah. And the working theory has always been that if you can just, scrub that biochemical debris out of the brain, you'll stop the cognitive decline.
Which makes sense on paper.
It does.
Oh.
And this is but what if we spent the last 25 years trying to fix a chemistry problem when Alzheimer's is actually, well, a geometry problem?
It completely upends the entire paradigm. I mean, it really does.
Right.
Because if you look at the clinical trial data over the last quarter century, the biological approach focusing purely on, clearing out plaques has yielded incredibly marginal results.
Barely moving the needle.
Exactly. We are actually very good at removing amyloid plaques from the brain at this point.
We are.
Yeah, we have monoclonal antibodies that do exactly that. They sweep the brain clean.
Wow, okay.
Yet the cognitive decline often just continues. The fundamental architecture of the neurons still fails, even with the plaques gone.
Which implies we are, looking at the smoke and entirely ignoring the fire.
Precisely.
And that realization brings us to the source material we are diving into today.
It's a fascinating piece of work.
It really is. It's a 2025 manuscript by George James Dukas, and it's titled The Transdimensional Unified Field Theory of Neurodegeneration, or TDUFT for short.
TDUFT.
Right. So our objective today, the mission for this deep dive, is to unpack exactly how this theory uses operator geometry and physics to explain Alzheimer's.
And not just explain it, but mathematically connect it to other diseases.
Yes. We're going to look at how it mathematically connects neurodegeneration to its ultimate biological opposite, which is cancer.
Which sounds crazy at first, but the math is wild.
Totally wild. And perhaps most importantly, we'll see how it uses this new geometric understanding to propose a radically different blueprint for an actual cure.
What makes this manuscript so compelling, right from the start, is that it moves the field away from phenomenology.
Phenomenology. Unpack that for me about.
Sure. Phenomenology is essentially just cataloging the downstream rubble.
Yeah.
You see a plaque, you label the plaque, you target the plaque.
You're just sweeping up the mess.
Exactly. But this theory argues that the plaques and tangles are just the inevitable downstream symptoms of a root geometric collapse.
Like the aftermath of an earthquake.
Right. It's looking at the upstream structural mathematics of why the building fell in the first place, rather than just sweeping up the bricks on the sidewalk.
Okay, let's unpack this core premise, because the shift from, wet biology to theoretical geometry is a heavy lift.
It's a huge shift in thinking.
Yeah. So before we can even talk about how this disease spreads or how to cure it, we really have to understand the fundamental structures that are failing in the Alzheimer's brain.
Right, the root cause.
And the manuscript introduces this concept of K-shells. Now, for someone who hears the word shell and thinks of, like, a seashell or maybe an artillery shell, what exactly is a K-shell in the context of our biology?
Well, to understand a K-shell, you really have to think about biology not as a random soup of chemicals just sort of bouncing into each other.
Which is how I usually picture it, honestly. Just a messy soup.
A lot of people do, but you have to see it as a highly organized mathematical structure. In the TDUFT framework, every biological function is governed by a specific geometric domain, and that domain is denoted by the letter K.
Okay, K for the domain.
Right. You can think of a K-shell as a localized physical and mathematical boundary that governs a very specific biological subsystem.
Kind of like a designated work zone.
Yeah, exactly. And within that boundary, every shell has two critical properties that absolutely dictate whether you are healthy or sick.
Yeah, those are.
Its torsion and its microstate limit.
Okay, torsion and microstate limit. Let's break those down because they are basically going to be the foundation for everything else we talk about today.
They are the core metrics of the whole theory.
Right, so let's start with torsion. What does that mean here?
So torsion, in physics and geometry, refers to the extent to which a curve or a structure deviates from being planar.
Planar meaning flat.
Right, flat. So torsion is a twisting force. It's a structural coherence. In biology, proteins have to fold into highly specific, complex, three-dimensional shapes to actually do their jobs.
They have to be twisted up in just the right way.
Exactly. They have to maintain a certain structural tension.
Yeah.
If they lose that torsion, if that structural coherence relaxes or collapses, the protein misfolds.
And then it's useless.
Worse than useless sometimes. It can no longer function at all.
It makes me think of, like a tightly wound rubber band on one of those little balsa wood model airplanes.
Oh, that's a perfect analogy.
Right, because the energy and the function of the propeller rely entirely on the torsion, the twist, of that rubber band. If it loses its tension and just goes slack, the whole system just stops working.
That's a great way to visualize it. The tension is the function.
Yeah.
And that brings us to the second property, the microstate limit.
Okay, microstates. Walk me through that.
So in thermodynamics, a microstate is a specific microscopic configuration of a system. For our purposes today, it's basically the number of possible geometric shapes or configurational states a shell can exist in without falling apart.
So it's like how many ways it can bend without breaking.
Precisely. It's a measure of structural tolerance. A higher microstate count means a shell can absorb more stress, it can shift into different configurations, and it can still maintain its overall integrity.
Okay, so a lower microstate count means the shell is more brittle.
Right.
It has way fewer options to adapt before it just completely snaps.
Exactly. And the manuscript identifies 3 specific, highly vulnerable K-shells that form the structural foundation of a healthy neuron.
Just 3.
Just 3 main ones for this specific cascade. And when these specific shells fail, that is what we diagnose in the clinic as Alzheimer's disease.
And these are K equals 6, 7, and 8.
Correct. K6, K7, and K8.
Let's walk through these one by one because I feel like this is where the theoretical geometry actually maps directly onto the hard biochemistry we've been hearing about for decades.
Yes, this is where the math meets the meat.
Love that. Let's start at the beginning of the cascade.
Yeah.
The K6 shell. What is happening there?
So the K6 shell is defined mathematically as the hydrophobic carbon shell.
Hydrophobic carbon.
Right. And in the physical, wet reality of the brain, this corresponds to the core stability of the amyloid beta protein.
Oh, okay, so this is the famous amyloid we always hear about.
The very same. Now, the manuscript calculates that this specific geometric shell is relatively fragile. It only has roughly 200 microstates.
Wait, 200? That sounds incredibly low.
It is. Specifically, the math notes an entropy value that yields about 199 functional states.
So that's a really low structural tolerance, like a very rigid rubber band.
It's highly constrained. And remember, it's a hydrophobic shell. Hydrophobic literally means it repels water. It hates water.
And the brain is, what, mostly water?
Exactly. It's an incredibly watery environment. So these hydrophobic carbon chains naturally want to fold inward, hiding away from the water, locking into a very specific geometric shape just to maintain stability.
They're trying to stay dry.
Right. But when the torsion coherence in this K6 shell drops below a critical threshold.
When our rubber band loses its tension.
Yes, exactly. When that happens, the hydrophobic carbon can no longer maintain its proper geometric packing.
Meaning it unfolds. It can't hide from the water anymore.
Exactly. The geometry completely destabilizes. The carbon structures become exposed to the aqueous environment, and because they are absolutely desperate to avoid the water, they start aggressively clumping together.
Clumping with what?
With other exposed hydrophobic carbons from other failing proteins nearby. They stick together to hide from the water.
Oh, wow. And that clumping.
Yeah.
That is what biologists call beta sheet aggregation,
Sure,
right?
got it.
Which eventually forms the massive amyloid plaques that we physically see on MRI brain scans.
Yes.
So in this geometric framework, the plaque isn't some toxic invader that just showed up out of nowhere to attack the brain.
Not at all.
It is the literal physical manifestation of a geometric collapse. It's just the rubble of the K6 shell falling apart.
That is the exact paradigm shift this paper is pushing. The plaque is the symptom of the torsion failure, not the root cause of the disease.
Okay, that makes so much sense. So that's K6. We have our amyloid core collapse.
Yeah.
The next shell in the sequence is K7, which the author calls the nitrogen torsion shell.
Yes. So if K6 is all about amyloid, K7 corresponds to the stability of the tau protein.
Tau, the other big buzzword in Alzheimer's.
Exactly. Specifically, it governs the microtubule binding repeats of the tau protein. Now, tau is an incredibly nitrogen-rich protein. This shell is structurally more complex than the amyloid shell.
So the math is different here.
Yeah, the math dictates it has roughly 540 microstates.
Okay, so 540. It has a bit more structural tolerance than the carbon shell, which was at 200, but it's still relatively vulnerable.
Well, currently still vulnerable.
So how does tau actually function in a healthy brain? And what happens when the portion fails at this specific shell?
Right. So picture a healthy neuron. Inside it, you have these long structural highways called microtubules.
Like train tracks.
Exactly like train tracks. They transport nutrients, organelles, and essential materials from the main cell body all the way down the long, spindly axon.
Okay.
Tau is the protein that acts like the wooden railroad ties holding those microtubule tracks together.
So it literally holds the highway intact.
Yes. Because of its specific nitrogen-rich geometry, it maintains a tight torsional grip on those microtubules. But when the torsion in the K7 shell fails, that structural twist is lost.
The rubber band goes slack again.
Right. And when that happens, the tau protein physically loses its grip.
It literally just lets go of the tracks. It lets go, it misfolds, and then it begins to tangle up with other free-floating tau proteins.
Oh, and that creates the famous neurofibrillary tangles.
Exactly. But honestly, the real devastation isn't just the tangles. It's what happens to the tracks themselves.
Because the railroad ties are gone.
Precisely. Without tau holding them together, the microtubules disintegrate. The neuron's entire internal transport system just completely collapses.
The cell essentially starves because it can't move supplies anymore.
Exactly.
Wow. So K6 is the amyloid plaques forming outside the main structures, and K7 is the structural highways collapsing inside the cell.
Right. Inside and out.
That is brutal. And that leaves us with the final shell in this triad, K8, the oxygen phosphorylation shell.
Yes. And this one is the most complex of the three. It operates with roughly 1,330 microstates.
Okay, 1330, so much more robust structurally.
It is, and it governs a massive system, the flux of kinases and phosphatases.
Kinases and phosphatases. Remind me what those do. These are the enzymes responsible for phosphorylation. That's the biological process of adding or removing phosphate groups to proteins.
Which is basically the on-off switch for almost every cellular function,
Well,
right?
exactly. It's how the cell controls what's happening at any given millisecond. And this whole system is driven by oxygen geometry.
Okay, so what happens when K8 goes down?
When a geometric failure hits the K8 shell, the regulation of this oxygen-based phosphorylation is just completely destroyed. The system goes into a massive overdrive.
Like a short circuit.
Exactly like a short circuit. The kinases go hyperactive. They start indiscriminately adding phosphate groups to everything in sight.
Just throwing switches wildly.
Yeah. And remember those now loose tau proteins from the K7 collapse?
The ones floating around making tangles.
Right. They get hyperphosphorylated by this K8 glitch, which actually makes them even more toxic. And eventually the synapses, the crucial communication bridges between neurons, they are just severely damaged by this unchecked chemical and electrical flux.
Man, if we visualize this whole thing as a massive architectural failure, it feels exactly like a sequence of structural tension giving way.
This structural nightmare.
Imagine a really complex suspension bridge, like the Golden Gate. K6 is the main suspension cable starting to stretch and sag, right? They lose their torsion, and the carbon steel starts to actually deform.
Right, the tension drops.
And because those main cables are sagging, all that physical stress has to go somewhere. So it transfers to the vertical suspender cables, the smaller ones holding the road.
Yes.
That's K7, or nitrogen shell. They weren't designed to hold that much primary tension, so they just violently snap.
They let go of the road.
Exactly. And once they snap, the actual roadway, the bridge, the K8 shell, where all the electrical and chemical traffic flows, it buckles and just collapses entirely into the river.
I mean, that is a phenomenal way to visualize the transfer of stress. It is, by definition, a cascading structural failure.
But I need to push back here for a second.
Go for it.
Because this all sounds incredibly elegant on paper. The math, the bridges, the rubber bands. But we are talking about microstates and geometric shells. And biology labs don't use protractors to diagnose Alzheimer's.
No, they certainly don't.
Right. They use chemical assays, they use microscopes, spectrometers. So how does a purely theoretical physics model actually ground itself in the messy, wet reality of biology? How do we know these specific mathematical K-shells correspond to real physical proteins?
That is honestly the most crucial question you could ask. And frankly, if the author hadn't addressed it, this paper would just be, mathematical poetry.
Right. Beautiful but useless for patients.
Exactly. But the manuscript is actually highly rigorous about providing operational biophysics definitions for every single shell. It ties the abstract geometry directly to standard lab measurable biophysics.
Okay, I need examples.
Ugh.
How do you actually measure a K6 torsion collapse in a physical lab?
You do it through structural biology tools that measure the physical shape of molecules. For the K6 hydrophobic collapse, the manuscript points specifically to circular dichroism spectroscopy and theoflavin T fluorescence.
Let's break those down because that sounds intense. What is circular dichroism spectroscopy?
Circular dichroism, or CD for short, is a technique where you shine circularly polarized light directly through a solution containing your protein.
Circularly polarized light, like the light waves are spinning.
Exactly. And because proteins are chiral, meaning they have a specific handedness or structural twist to them, they will actually absorb right-handed spinning light and left-handed spinning light differently.
Depending on their shape.
Yes, depending on their exact three-dimensional shape. If a protein is in its healthy, twisted state, you get one specific absorption signature on the graph.
Okay.
But if it collapses into a flat beta sheet, which, as we discussed, is what happens when K6 fails, the absorption signature changes dramatically.
Oh, wow. So CD spectroscopy physically measures the loss of that geometric twist.
It does. It gives you a literal graph of the torsion failing.
So you are literally using the rotation of light to measure the torsion of the protein. That is amazing.
It's brilliant.
And what about theoflavin T fluorescence?
Theoflavin T is basically a chemical dye. It behaves completely normally when it's just floating in water, but when it physically binds to the specific repetitive geometric structure of those amyloid beta sheets we talked about.
Oh, plaques.
Yes, the plaques. When it hits that specific collapse geometry, it suddenly fluoresces incredibly brightly under a microscope.
It lights up like a Christmas tree.
Exactly. It is a direct visual laboratory confirmation of the K6 geometric collapse.
Okay, so K6 is highly measurable. We can see it. What about K7, the nitrogen torsion loss? How do you measure a microscopic tau protein losing its grip on a microtubule?
For K7, the author points to two things: NMR, order parameters, and hydrogen deuterium exchange mass spectrometry, or HDXMS.
Okay, NMR, nuclear magnetic resonance. It's basically like an MRI for molecules, right?
Exactly like an MRI. Using NMR, specifically looking at what we call residual dipolar couplings, you can actually measure the average orientation of specific chemical bonds relative to a magnetic field.
Okay, so what does that tell you?
It tells you exactly how rigid or floppy a specific part of a protein is. You can mathematically calculate the loss of torsional order just by seeing the bonds become less constrained, more wobbly.
So you can literally see the structural integrity dropping.
Yes, and HGXMS is even more elegant, in my opinion.
How so?
Well, you basically bathe the tau protein in heavy water.
Heavy water, like radioactive.
No, not radioactive. Just water where the normal hydrogen atoms have been replaced by deuterium, which is just a heavier, stable isotope of hydrogen.
Oh, okay. So you soak the tau protein in this heavy water. Then what?
Well, the heavy deuterium in the water will slowly swap places with the normal hydrogen atoms on the surface of the protein.
Just a natural chemical exchange.
That's right. And here's the key. It can only swap with hydrogens that are physically exposed to the water. Exactly. If the protein is tightly wound, holding onto that microtubule, a lot of its hydrogens are hidden and protected on the inside.
They stay dry.
Right. But when the K7 torsion fails and the protein unfolds and lets go, all those previously hidden nitrogen-hydrogen bonds are suddenly exposed.
And they get hit by the heavy water.
Yes. They rapidly take up the heavy deuterium. So when you pull the protein out and weigh it in a mass spectrometer, it's suddenly heavier.
Wow.
The added weight is a direct, quantifiable measurement of its structural unraveling. You are weighing the loss of geometry.
That is so incredibly clever. You are literally weighing the collapse. Okay, and for K8, the phosphorylation overload.
That is measured by targeted mass spectrometry as well, specifically mapping the exact stoichiometry of the phosphate group.
Stoichiometry meaning like the counting of them.
Yes, you can literally count the hyperactive addition of oxygen-heavy phosphate molecules to the protein.
So it's a measurable frenzy of activity.
Exactly. The point is, the geometry translates perfectly into the hard empirical data sitting on a biochemist's desk. The math predicts a structural failure, and the physical instruments confirm exactly that structural failure.
Okay, well, that completely satisfies my skepticism on the tangibility of the math. We know what fails: the K6 amyloid core, the K7 tau microtubule bridge, and the K8 phosphorylation regulator.
Right.
But, knowing what fails doesn't really explain the disease itself.
How do you mean?
Well, if a single bridge cable snaps, the bridge doesn't necessarily fall. It might sag. But Alzheimer's spreads. It progresses deterministically like a slow-moving fire through the entire brain. Why does it do that?
And that brings us to the core mechanism of the manuscript. the propagation of the disease. The author defines Alzheimer's mathematically as a forward-propagating torsion collapse.
Forward propagating, meaning it always moves in a specific sequence, a specific direction.
Yes, always. The theory states that the overall health of any given synapse is dependent on a minimum combined torsion threshold of these three shells.
A combined threat.
Right. If the combined structural integrity of K6, 7, and 8 falls below a critical minimum, the synapse just dies. But the key insight of this paper is that they don't fail independently and they don't fail randomly.
They fail sequentially.
Yes, due to a thermodynamic mechanism the author calls a coupling.
A coupling. Okay, so the K is the shell, like we established. What is the A?
K refers to the distance between the shells. In this geometric framework, biological shells influence each other based on their numerical distance.
Oh, interesting.
The closer they are in the numerical sequence, the more powerfully their geometries interact. So the coupling between K6 and K7 is incredibly strong because their act distance is only 1.
They are literally adjacent architectural systems.
Exactly.
Which means if something happens to one, the other feels it immediately, like a shock wave.
Yes, and to understand why one actively forces the other to fail, we have to look at the thermodynamics of configurational entropy.
Oh boy. Okay, let's unpack configurational entropy, because that sounds like a term that gets thrown around in physics lectures a lot. But what does it actually mean for a wet protein in my brain?
It's actually simpler than it sounds. Remember when we talked about microstates? The number of geometric shapes a protein can take without breaking.
Right, the 200 states for amyloid, 540 for tau.
Exactly. Configurational entropy is essentially just the measure of those microstates. It's the energy stored in the structural flexibility of the shell.
The bounce in the spring.
Yes. When a shell is healthy, it has high entropy. It has many functional configurations. It can bounce around. But when the torsion fails, the shell collapses into a single, rigid, dysfunctional state.
Like the amyloid beta sheet.
Right.
It goes from being a flexible spring to basically a rigid piece of plastic.
Precisely. And thermodynamics demands that the energy that was stored in that previously flexible spring has to go somewhere.
Because energy cannot be destroyed.
Exactly. First law of thermodynamics. So when the K6 microstate count collapses, it violently sheds its configurational energy into the surrounding environment.
And because K7 is sitting right next door, structurally speaking, it takes the brunt of that energy wave.
Exactly. It gets hit hard. The shed energy from the K6 collapse radically alters the localized physical environment of the K7 shell.
So it makes it unstable.
Yes. It effectively lowers the energy barrier required for the tau protein to lose its own torsion. The amyloid collapse actively destabilizes its neighbor. It forces the tau protein to let go of the microtubule.
It pushes it over the edge.
Yeah.
And once K7 collapses and lets go, it sheds its energy, which then hits the next neighbor in the sequence, K8, the phosphorylation shell.
Exactly. And because K8 governs the high-energy processes of those kinases, when it receives that massive destabilizing shock wave, it doesn't just shut down.
You short circuit.
It short-circuits into massive overactivity.
Wow. It's exactly like the suspension bridge analogy we were just talking about. When the main K6 cables stretch and fail, the structural stress doesn't just vanish into thin air.
It can't.
Right, it transfers. It bleeds directly into the K7 suspender cables. The suspender cables aren't built for that specific geometric load, so they snap, transferring an impossible load onto the K8 roadway, which then buckles.
It's a deterministic cascade.
Completely deterministic. Once the forward propagation begins, thermodynamics dictates the sequence. 6 forces 7 to fail. 7 forces 8 to fail.
And this right here explains so much about the utter frustration in Alzheimer's research over the last 20 years.
Right. If you look at the history of these clinical trials, we've spent 2 decades engineering drugs that act like, highly specialized welders to go in and fix the K6 main cables.
We clear out the amyloid plaques. We weld the cables.
But if the K6 cables have already transferred their failure to the K7 suspender cables and the K7 cables have already snapped, fixing the main cable doesn't matter anymore.
Not at all. The roadway is already falling.
The roadway is falling. That is exactly the tragedy of single-target amyloid therapies. They are trying to arrest a forward-propagating collapse by fixing a domino that has already fallen.
And transferred its momentum to the next domino in the line. The cascade has simply moved on.
Man. Which provides basically the perfect transition into exploring how this theoretical framework views the broader landscape of disease.
It's a much bigger picture than just the brain.
Yeah, because if Alzheimer's is a forward-propagating structural collapse, what does this math say about other diseases? To truly understand the nature of this mechanism, the author contrasts it with the ultimate biological opposite of degeneration,
Uncontrolled growth.
cancer.
This is where the manuscript elevates from being just a paper about neurodegeneration to essentially a unified field theory of pathology.
A theory of everything for disease.
Right. The author compares the Alzheimer's mechanism directly to a universal cancer theory, which was derived using the exact same operator algebra.
So they use the exact same geometric math to explain both Alzheimer's and cancer.
Yes. In the TDUFT framework, the equations governing Alzheimer's and cancer are identical. They use the same K-shells, the same thermodynamic principles of microstates, and the same torsion mechanics.
But they manifest so differently.
That's because they are categorically different in their geometric direction and the fundamental nature of their endpoint.
Okay, right. Alzheimer's is defined as a collapse-type disorder. It moves forward sequentially. 6, then 7, then 8. The structural tension fails, the microstates drop, and it ends in a collapsed, dying neuron.
Right. But cancer is defined mathematically as a vacancy-type disorder, and it moves backward.
Backward. So how does that sequence look?
The progression for cancer in this mathematical model is a reverse sequence. It originates around K7, moves backward to K8, and its ultimate terminal endpoint is K11.
Like K11, we haven't talked about that shell yet. What does K11 do?
The K11 shell is the division control shell.
Division control, like cell division.
Exactly. In cellular biology, this corresponds to the G1S pathway.
Which is what exactly?
It's basically the checkpoint that regulates whether a cell is allowed to divide and replicate its DNA. In a healthy cell, the K11 shell maintains strict geometric control over this checkpoint. It keeps the door locked until it's time.
So what happens when a vacancy-type defect hits K11? Does it collapse?
No, unlike Alzheimer's, which ends in a heavy, rigid structural collapse, cancer ends in a missing operator microstate.
I see.
It is literally a geometric vacancy at K11. It's not that the structure collapsed under tension, it's that a crucial regulatory lock has simply vanished.
Oh, wow. So going back to our structural engineering metaphors, Alzheimer's is the suspension bridge collapsing under its own failing tension. The materials give out and the structure falls into the river.
Right.
But cancer is a highway where someone has completely removed the toll gate entirely. There is no structural collapse. There is just an absolute absence of a regulatory boundary.
So the traffic just floods through.
The traffic of cell division floods through unchecked forever.
That is a brilliant way to distinguish them. One is a failure of structural tension, ending in death. The other is the catastrophic absence of a boundary, ending in uncontrolled replication.
But I got to play devil's advocate for a moment here.
These do.
Because anytime you have a unified theory that connects two completely different diseases, alarm bells start ringing for me.
Naturally.
If cancer propagates sequentially, say 7, 8, 11, and Alzheimer's propagates sequentially 6, 7, 8, they share shells. They both interact heavily with 7 and 8.
You do cross paths there, yes.
So does that mean they exist on the same spectrum? Like, if I develop a drug to drastically alter the torsion of the K7 tau shell to fix a patient's Alzheimer's, is there a risk that I accidentally initiate a backward propagation and cause a K11 vacancy? Could treating neurodegeneration accidentally cause a tumor?
It's a very logical, even necessary concern when you are dealing with unified biological mathematics. You have to ask that question.
Right.
But the mathematics of the manuscript strictly prohibit that outcome.
Why? How does the math protect against it? It's the same shell.
Because the defect types are fundamentally opposed in their energetic signatures. In the equations, cancer is represented by a negative change in the overall microstate space. It's denoted mathematically as delta omega is less than 0.
Meaning a loss of states.
It is a thermodynamic subtraction. It requires a specific energy environment to carve out that vacancy.
Okay.
Alzheimer's, conversely, represents a positive accumulation of rigid states. It is a collapse into an overloaded, hyperdense structural knot.
So one is a thermodynamic sinkhole and the other is a thermodynamic mountain.
Precisely. Because the defect types are mathematically inverted like that, you cannot accidentally cause a vacancy while applying the specific geometric pressure required to stabilize a collapse.
So the tool for one literally can't do the job of the other.
Exactly. A molecule designed to push against a collapse simply does not possess the structural capacity to carve out a vacancy. They are mutually exclusive geometric interventions.
Okay, that makes sense. Which brings us to arguably the most exciting part of this entire deep dive. Section 4 of the manuscript.
Yes. Engineering the cure.
Engineering the geometric cure. Having established this incredibly precise, mathematically verified mechanism for how the disease actually works, the text naturally transitions into how we build a physical molecule to invert this specific math.
And it's not just a vague conceptual idea.
No. The author provides a highly specific blueprint.
Yes. Because Alzheimer's is a forward-propagating torsion collapse across 3 distinct shells, a cure cannot just be a single target antibody.
It can't just be the K6 welder.
Right. It can't just fill a vacancy either. To stop the cascade, the proposed molecule must act as a 3-shell torsion stabilizer.
3 shells at once.
It must physically bind to and structurally support K6, K7, and K8 simultaneously.
It basically has to brace the entire failing structure all at once. And the author gives us the exact molecular formula for this biological brace, which is C14H22 and 2O3.
That's the one.
It has a molecular weight of 264 Daltons. Let's start right there. A little late, because I know that number is incredibly important in neuroscience. Why 264 Daltons?
The biggest physical hurdle in treating any brain disease is the blood-brain barrier, or BBB.
Right, the brain's bouncer.
Exactly. It's a highly selective, semi-permeable border that protects the brain from toxins and pathogens circulating in the blood. But unfortunately, it also blocks most therapeutic drugs.
It does its job too well.
The general rule of thumb in neuropharmacology is that a molecule must be under 400 Daltons to easily slip through that barrier without help.
Under 400. And most of the monoclonal antibodies we currently use to clear amyloid plaques are what?
Oh, they are massive. They are tens of thousands of Daltons.
Why?
They have to be infused in massive quantities intravenously just to get a tiny, tiny fraction across the barrier.
But this engineered molecule, C14H20N2O3, is small. At 264 Daltons, it is perfectly sized for passive diffusion across the blood-brain barrier.
It doesn't need to be forced. It naturally slips right in.
Okay, so it gets into the brain. Great. Now it has to actually do the work. The molecule requires 3 distinct geometric modules to match the 6 to 7 to 8 collapse pathway. Let's break down the chemistry of this blueprint piece by piece, starting with the base. The K6 scaffold.
So the manuscript proposes a phenyl cyclohexyl backbone for the K6 scaffold. This specific chemical structure is carbon-rich and highly hydrophobic.
Which perfectly matches the hydrophobic carbon nature of the K6 shell we talked about earlier.
Exactly. Form follows function. Its job is to serve as the foundational anchor. Because it is highly hydrophobic, it gets drawn directly to the collapsing amyloid beta sheets, which, remember, are also desperately trying to hide from water.
Like magnets.
Yes. It physically interlocks with the K6 shell and provides the structural rigidity needed to stop that initial hydrophobic collapse.
In our suspension bridge analogy, this is the massive steel clamp that you bolt onto the failing main cable to stop it from sagging any further.
Yes, that's the anchor. But as we know, fixing the main cable isn't enough if the disease has already propagated. So covalently bonded to that phenyl cyclohexyl backbone, you have the K7 torsion bridge.
And the text specifies this as an amide, or NH linker.
Right. It's a nitrogen-rich module. This geometrically aligns with the nitrogen-rich tau protein in the K7 shell.
Okay.
Its function is to physically bridge the gap and restore the microtubule binding torsion. It provides the artificial tension required for tau to grip the cellular highways again.
So the scaffold anchors to the amyloid, and the bridge reaches over and braces the snapping suspender cables of the tau protein.
Exactly.
And finally, sitting on top of that bridge is the K8 regulator.
The author suggests a hydroxyl or O-methyl head group for the regulator. This is an oxygen-based chemical structure which corresponds directly to the oxygen-driven phosphorylation shell at K8.
Okay, so it matches the elements.
Yes, and its specific geometry is designed to interact with the kinase and phosphatase enzymes. It physically blocks the hyperactive phosphorylation, effectively cooling down the overloaded system.
Wow. So you have a single, small molecule that enters the brain, anchors to the amyloid, braces the tau, and calms the enzymes. It stops the entire 678 cascade simultaneously.
That's the design.
But whenever you introduce a molecule that interacts with fundamental cellular structures, you immediately run into the problem of toxicity.
Always.
How does this drug avoid binding to healthy proteins and completely disrupting the normal function of a healthy neuron?
This is where the geometric approach shows its true elegance, honestly. The manuscript addresses side effects through what it calls the shell distance constraint.
Shell distance constraint.
In traditional pharmacology, you usually rely on chemical affinity to target a drug, basically just hoping it prefers the sick cells over the healthy ones.
Right, which leads to horrible side effects.
Exactly, but the TDUFT framework solves this geometrically.
How so?
The drug is engineered with a strict mathematical requirement. Its total equidistance must be greater than or equal to 5 relative to healthy tissue.
Meaning its geometry is mathematically separated from the geometry of a healthy shell.
Precisely. Think about a healthy K 6, 7, and 8 sequence. In a healthy neuron, those shells have high torsion coherence. They are tightly wound, structurally robust, and geometrically perfect.
Yeah.
The C14H20N2O3 molecule is specifically built to fit the looped, elongated, collapsed geometry of the failing Alzheimer's shells.
So if this drug floats by a healthy protein,
It literally bounces off.
it bounces off.
It is geometrically incompatible. Imagine trying to force a strangely shaped puzzle piece into a section of the puzzle that is already perfectly assembled.
It won't fit.
It just slides right off. The molecule can only bind to the structural voids that are created by a torsion collapse.
The level of engineering discipline in this text is just astounding to me. It bridges theoretical cosmology and practical medicinal chemistry in a way I've really rarely seen.
It's a very complete blueprint.
It even outlines the synthesis route, right?
Right.
Like detailing the reductive amination, the amide coupling, the hydroxylation required to actually build the molecule in a wet lab.
Yes, the chemistry is all there.
And it dictates strict pharmacokinetic parameters, like aiming for a log P of 1.2.
Ah, that log P value is another crucial detail.
Unpack log P for me.
Log P is basically a measure of a molecule's lipophilicity, meaning how well it dissolves in fats versus how well it dissolves in water.
Okay.
A log P of 1.2 is basically the absolute sweet spot for a central nervous system drug. It's lipid-soluble enough to cross the fatty membranes of the blood-brain barrier, but it's water-soluble enough to travel freely through the aqueous fluid of the brain to actually reach the neurons.
It is an engineered, bench-ready blueprint.
It is.
And here's the crux of science, as any good scientist knows. A theory is only as valid as its ability to be proven wrong in the real world.
Absolutely.
You can build the most elegant mathematical model in the history of the world, but if it doesn't hold up in a clinical trial, it's useless. And the author clearly understands this because the manuscript transitions into a really rigorous section on falsification.
The willingness to subject the theory to falsification is, to me, the hallmark of rigorous science. The author doesn't just propose the molecule. They outline 6 strict criteria labeled P1 through P6.
Right.
And they state, unequivocally, if experimental data fails to confirm any of these 6 predictions, the entire geometric model must be rejected.
You literally have to provide the gates for your own destruction.
You do.
Let's look at some of these specific predictions. P1 is fascinating to me. It states that torsion precedes phosphorylation. What does that mean for a lab researcher testing this?
It predicts a strict unidirectional timeline of biological events. P1 states that if you introduce a drug that specifically restores the K7 torsion, if you brace the tau structure, you will inevitably observe a reduction in tau misfolding before any change in the K8 phosphorylation occurs.
Why is that a radical claim?
Because for a long time, the prevailing thought in the field was the opposite, that rogue enzymes caused hyperphosphorylation first, which then caused tau to misfold.
Ah, but this geometric model says no.
Right. It says the structural failure happens first, which then triggers the enzymatic overload.
So if a lab tracks this in real time,
Using iPSC-derived neurons, for example, neurons grown in a dish from human stem cells, and they see phosphorylation changing before the structural twist changes. The theory is dead.
wow, that is a highly testable claim.
Very testable.
Then we have P2, the threshold dependence. And frankly, this point alone explains so much about modern pharmaceutical failures. Can you explain the P2 prediction?
Sure. P2 deals with the critical minimum threshold for synaptic viability that we talked about. It states mathematically that if a patient's overall structural coherence is already past the hyperphosphorylation threshold, meaning the K8 shell is already in full cascade, a drug that only targets the upstream K6 amyloid shell will mathematically fail to restore the synapse.
Because the transfer of failure has already occurred. The domino has fallen.
Exactly. P2 predicts that single-target amyloid clearance in late-stage progression is thermodynamically useless.
And if you look at the clinical data from the last decade, that prediction mirrors reality perfectly.
Sadly, it does.
We clear the amyloid, but the patients don't recover their memory because K8 is already collapsing. The math explicitly requires a multi-shell intervention once that threshold is crossed.
It provides a perfectly logical thermodynamic explanation for why those billions of dollars in trials yielded such marginal results.
It's sobering. And then there is P4, the PT propagation law. This one moves from the cellular level to the macro level of neuroimaging, right?
Yes. Currently, we use PT scans, positron emission tomography, to visualize the physical spread of amyloid plaques in a living brain.
Right, you can see the glowing spots on the skin.
Exactly. And the traditional assumption is that the density of the plaque predicts the speed of the disease.
More plaque, faster decline.
Right. But P4 challenges that. It states that the static load of amyloid doesn't actually predict disease velocity. Instead, the rate of change in the torsion of K6 and K7 is the true predictor of how fast the PT signals will spread.
So it's not about how much garbage is on the floor, it's about the mathematical rate at which the ceiling is currently cracking.
That's a great analogy. If a researcher can design a PET tracer that binds to torsional state rather than just protein mass, and that tracer fails to predict the spread better than a traditional amyloid tracer, the theory is falsified.
It is offering a completely testable alternative to the current imaging paradigm.
It is. It is incredibly rare to see a theoretical physics paper willingly subject itself to organoids, mass spectrometry, and clinical neuroimaging standards.
But what's crazy is that the ambition of the manuscript doesn't even stop at Alzheimer's.
No, it zooms way out.
In fact, it zooms out to contextualize Alzheimer's within a much larger, completely comprehensive system. And this brings us to the Shell periodic table.
Yes, the Shell Index Disease Taxonomy, or SIDT. This
Right.
forms the foundation of what the author calls unified Shell-level medicine.
Just as Mendeleev looked at the chaotic mess of chemical reactions and realized they could all be organized into a periodic table based on atomic structure and electron shells, this theory proposes a periodic table of human disease based on these biological K-shells.
That is the exact ambition of the text. The manuscript outlines 12 biological shells in total, categorized by increasing microstate complexity.
We've spent this entire time basically talking about the cascade of 6, 7, and 8 for Alzheimer's, and the vacancy at 11 for cancer.
Right.
But this table fills in the rest of human pathology.
It does.
Let's explore this map a bit. What else is on the table?
Okay, so shell K4 is defined by sulfur geometry. The theory posits that this shell governs mitochondrial function.
Okay, the powerhouse of the cell.
Right. Therefore, diseases categorized by profound energy deficits, mitochondrial collapse disorders, are mathematically defined as K4 defects.
Okay, moving up the complexity scale, what about K5?
Shell K5 is governed by aromatic geometry structures involving carbon rings. And the table makes a very specific prediction here. It defines Parkinson's disease as a 5 to 6 to 7 collapse.
Wait, so Parkinson's is just a shifted sequence. It's the same mechanics, just starting one shell lower.
Exactly. The structural failure initiates at the aromatic shell, which governs structures related to dopamine production.
Which is exactly what fails in Parkinson's.
Right. And that collapse bleeds its entropy forward into the amyloid and tau shells.
No.
That explains why many late-stage Parkinson's patients eventually develop amyloid and tau pathologies that look remarkably similar to Alzheimer's.
Oh, wow. The cascade just started one step earlier in this sequence.
It's all connected.
That is fascinating. And what about ALS, or Lou Gehrig's disease?
ALS, along with frontotemporal dementia, are mapped as primary failures initiating directly at K7 and K8. They basically bypass the K6 amyloid shell entirely.
Which aligns perfectly with biological reality, right? Because ALS is heavily driven by catastrophic motor neuron transport failure. The K7 microtubule highway is breaking down,
Right.
but without massive amyloid plaque involvement.
Exactly. The math reflects the clinical presentation.
And then, as we discussed, way down at the end of the complexity scale at K11, you have the division control shell, where cancer emerges from a geometric vacancy. It literally creates a coordinate system for human disease.
It does. Instead of viewing diseases as isolated random accidents of biology, they are mapped as predictable thermodynamic pathways on a unified geometric grid.
Okay, I have to stop you because my skeptical alarm is basically deafening right now.
I get it.
This one single theory is claiming to mathematically explain literally everything from severe acidosis at K1 to mitochondrial diseases at K4 to Parkinson's, Alzheimer's, ALS, and all the way up to cancer at K11.
Yes, that is the claim.
Isn't this the literal definition of too good to be true? I mean, a single master theory of all disease feels like an academic overreach. It sounds like a panacea.
That is a very important distinction to make, and it brings up the danger of reductionism in science. We have to look very carefully at what the theory is actually claiming.
Okay.
It does not reduce biology to a single magical cure. It reduces it to a single algebra.
Okay, unpack that distinction for me. What is the difference?
Let's go back to your Mendeleev analogy, the periodic table. Before the periodic table, chemistry was a chaotic mess of trial and error. People were just mixing things hoping they wouldn't explode.
Alchemy,
Pretty
basically.
much. But when the periodic table was introduced, it didn't make chemistry too simple. It didn't offer one universal chemical that did everything.
True.
It didn't mean that making table salt was the exact same physical process as making nitroglycerin.
Right, they are totally different.
What it did was organize the chaos into predictable, foundational rules based on electron shells. It provided a unified framework for understanding why chemicals reacted.
Yes, there you go.
This theory is attempting to do the exact same thing for pathology. It maps the chaos of biological disease into predictable geometric defect currents.
So it's not a unified pill, it's a unified language.
Exactly. A K4 mitochondrial disease still requires a totally different, highly specific custom-engineered molecule than a K11 cancer defect. You can't use the C14H20N203 Alzheimer's pill to cure Parkinson's because Parkinson's starts at K5.
The geometry is different.
It's not magic. It's just an argument that all of these disparate diseases are governed by the same underlying rules of geometric torsion and microstate thermodynamics. It's a blueprint for engineering custom repairs, not a singular cure-all.
That distinction makes a lot of sense. It provides the coordinate system for the engineers to work within.
Exactly.
Now, we've gone from biochemistry to microstate thermodynamics all the way to a periodic table of human disease. But in the final section of this manuscript, the author takes one last, massive conceptual step back.
It's quite a leap.
It is. We move from biology to cosmology. Because if human biology follows this incredibly strict, unified geometric algebra, what is that math actually derived from?
This is where TDUFT reveals its roots as a true unified field theory. The author posits a cosmological extension, demonstrating that the mathematical operators governing these biological K-shells map directly, identically, to the cosmological operators that govern the geometry of the universe itself.
So the math doesn't change when you scale it up.
The math remains perfectly consistent. According to the manuscript, K6, our hydrophobic amyloid shell, maps directly to what cosmologists call time packing.
Fly backing. Okay.
K7, the nitrogen torsion shell holding our microtubules together, maps identically to the velocity dihedral mode. And K8, our oxygen phosphorylation regulator, maps to the time flux mode of spacetime.
And the implications of this mapping are where the math gets truly mind-bending. Because the equations show that collapse defects, like the forward-propagating 6, 7, 8 cascade of Alzheimer's we've been discussing, mathematically correspond to positive curvature distortions in the spacetime field.
Yes, a structural knot, a high-density collapse.
While vacancy defects like the missing K11 tollgate that unleashes cancer correspond identically to negative curvature distortions in spacetime.
Now, we must tread carefully here as scientific interpreters.
Of course.
The text frames this as a mathematical and geometric unification, not necessarily a literal physical claim that a misfolded protein in your brain is generating a localized gravitational distortion field.
Right. It's not saying Alzheimer's is literally a tiny black hole in your head.
Exactly. However, mathematically speaking, the equations used to resolve the defect are indistinguishable. Treating a patient's Alzheimer's with that specific 678 stabilizing molecule is structurally and mathematically identical to a cosmologist applying a compensation operator to smooth out a positive curvature distortion in a theoretical spacetime field.
That is just, wow.
The algebra simply doesn't care if it is operating on a microscopic protein inside a human cell or the macroscopic fabric of a galaxy. The rules of geometric torsion apply universally.
I want you to really think about the gravity of that idea. The exact same mathematical laws governing the expansion, tension, and curvature of the universe itself might be the exact same laws governing how a protein folds in your brain to store a memory.
It implies a profound fractal nature to reality. The macrocosm of spacetime and the microcosm of human biology are bound by the exact same geometric constraints.
We are not separate from the physics of the universe. Yeah. We are a localized, highly complex expression of it. Let's try to pull all of these massive threads together and summarize the journey we've just taken through this manuscript. We started with what we thought we knew. Plaques, tangles, the biological rubble that we've spent 20 years trying to sweep out of the brain with massive single-target antibodies.
We observed the failure of phenomenology.
Then we shifted the lens. We realized those claques might actually be the downstream result of a geometric torsion collapse across 3 distinct biological shells: the K6 hydrophobic core, the K7 nitrogen bridge, and the K8 oxygen regulator.
Right.
We mapped how thermodynamics forces this collapse to propagate sequentially, bleeding energy from one shell to the next, like snapping cables on a suspension bridge.
We established the deterministic physics of the cascade.
From there, we examine the exact blueprint for a molecule C14H20N2O3, perfectly sized at 264 Daltons, to cross the blood-brain barrier designed to physically anchor to the amyloid, brace the tau, and calm the enzymes, stopping the entire collapse simultaneously while geometrically bouncing off healthy tissue.
A masterpiece of geometric engineering.
We looked at the strict rules of falsification required to prove it. And finally, we zoomed all the way out to see how this framework creates a unified periodic table of disease, seamlessly mapping human pathology onto the geometric curvature of space-time itself.
It is a staggering intellectual achievement, honestly, transitioning us from the messy guesswork of biochemistry to absolute structural mathematical precision.
So what does this all mean for the future of medicine?
I think it means everything.
It means we might be standing on the absolute edge of a medical paradigm shift. If this theory holds up to the rigorous lab testing it demands, we are moving away from a world of trial and error pharmacology, throwing isolated chemicals at downstream symptoms and hoping they stick.
We are moving into an era of precise geometric molecular engineering.
An era where we can calculate the exact mathematical shape of a disease and simply print the specific geometric inverse required to balance the equation and cure it.
And if we extrapolate that idea to its ultimate natural conclusion, it leaves us with a truly profound realization about ourselves.
Yeah.
If every affliction, from the rapid division of cancer to the slow degradation of Parkinson's and Alzheimer's, is ultimately just a defect in a specific mathematical shell sequence, then the human body isn't just a fragile bag of organic chemistry waiting to break down.
What is it then?
It is a localized, highly ordered, self-correcting mathematical field.
A localized mathematical field.
And if that is fundamentally true, it implies that aging, degeneration, and disease are not inevitable biological destinies that we just have to accept. They are simply imbalanced geometric equations waiting for the right inverse molecule to resolve them.
It reframes the entire concept of mortality.
It does. What happens to the trajectory of human civilization when we finally learn how to balance the master equation?
It makes you look at that muddy water of a medical diagnosis in a completely new light. It's not a broken plumbing system anymore. It's just a temporary tear in the geometry of the map itself. We just needed to learn how to read the coordinates.